Okay, after that somber introduction, I will try to lighten the mood a little bit as best I can by speaking about geometry and genetics. I will lean more on the biological phenomena and less on the equations and formalism, because if you're mathematicians, you could fill in the blanks. But it's important to focus on the natural science, which is biology. Okay, so I will talk about the phenomenon of canalization, which is basically that developmental outcomes are channeled into a few buckets, so to speak. I will talk about how mathematics embodies a certain phenomenology that is very concordant with development, speak about dynamics as geometry, and then talk about some examples with stem cells and development. Okay, so for canalization. The subject sort of begins with this book from 1894 from Bateman, which is basically a 500-page compilation of various oddities where, for example, a leg is transmuted to a wing or thumb to fingers. And the thing to note here is that the leg and the wing and the fingers are rather well formed. In other words, the outcomes are biologically recognizable structures that are substituted one for the other, which Bateman gave the name homeotic transformation, since back then biologists knew Greek, which they no longer do. A second high point is this famous experiment of Mangold-Spemann, in which they took a particular piece of a developing frog embryo from what will be the dorsal side. They transplanted it to the ventral side of a stage-match embryo, and lo and behold, you got a duplicated anterior axis. And note that with good technique, the two halves of the axis look rather symmetrical, i.e. you have two heads and a common gut. The phenomena is called induction because not only did this piece of transplanted tissue give rise to this, but it induced the surrounding tissue of the host to adopt the anterior fate and morphology. Very surprising. Another example, which particularly fascinates me, is regeneration. A salamander has the capacity which, when you lop off, even in the adult, a leg, that leg will regrow to the proper size. And if the salamander is growing, the leg will regrow to the then appropriate size of that salamander. You could have a trick system and get these ectopic forelimbs. And the trick here consists, it's rather interesting, consists of wounding the anterior side of the leg, bathing the animal in the posterior morphogen, and that confrontation of anterior and posterior morphogens gives rise to an ectopic leg forelimb. That is very surprising, has not been studied in sort of mechanistic detail, but for those interested in pattern formation, that is indeed a very striking phenomena and deserves to be better studied by people who are sort of model-based, model-centric. The same phenomena has long been seen in fly. So this is a duplicated a fly leg where the distal part has been duplicated with an extra pair of legs, or feet, so to speak, if I could say that. And part of the problem here, this is a rather old phenomena, and part of the issue is that you have to realize that the fly leg begins as a disc on the body with tens of cells. And you naturally ask, how does a disc turn into a leg? Of course, there's very substantial morphogenesis going on. And also, the disc has to decide. So the disc has morphogens that define anterior, posterior, dorsal, ventral. But the disc also has to define proximal distal. And it does so via this mechanism that was first postulated by Hans Meinhardt in the 40s, whereby you have three morphogens, and the center point will become where they overlap will become the most distal part of the leg. And how that works out, how this phenomenon works out to give you duplicated legs, is something which, again, is technically tractable these days, but no one's really doing the experiments because of the fads and fashions of fly developmental biologists that Rick could expound to us. But again, there's a very interesting problem there. Last situation is one that I was somewhat involved with. The patterning in the mouse embryo has been schematized as a disc by this eminent biologist. And some decade later or so, we started using stem cells to replicate those patterns. And I posit this as an instance of canalization because the topological arrangement of these layers precisely matches what was postulated by Tam as a representation of mouse gastrulation. And the interesting thing here is that all the morphogens, we know in some detail how this works molecularly, and the very elaborate cascades of activators, secreted inhibitors, and all the rest that operates in the embryo also operates in these patterns, which is a very surprising transposition of molecules to make patterns in a very different circumstance. Okay, so this all tells us, to bring up this cliche of an image, that we want to look at these decision points where we see a fork in the road and not worry about how to make a wing in detail, but worry about how we bifurcate from the leg fate to the wing fate. Okay, so just in case you wanted to be more pure and gene-centric, that is hopeless, following ideas of Hegel, in fact. This is the simplified, so there are roughly 10 pathways that suffice to guide all of vertebrate development and, in fact, also arthropod development. And Wnt is one of those 10 pathways. This is the simplified view of the Wnt pathway at the gene level, Wnt for WIMPs. You will not reduce that to a bunch of differential equations. You will die. So phenomenology is essential because you can't describe that in detail. Okay, so how do signals induce fates? If I could write down the kinetic equations for all those genes, and they are incomprehensible to anybody, biologist or mathematician. We want something that's much more like Pauling's theory of the chemical bond to make sense of that complexity, i.e. phenomenology. And the mathematics I will show you is sort of modern in that the protagonists are, I think, still alive, and is a very good instance of this quote from Dennis Sullivan that that very good mathematics, which I am not capable of doing, consists in making the right definition or concept such that what you want to prove is obvious. It's a bit flippant, as is appropriate from Dennis Sullivan, but is actually very apt. And perhaps it was the only thing of Sullivan that I understood at that meeting. Okay, so the deal is that you want to turn flows into landscapes. So on the left, you see a vector field. There is a source, there is a sink, and of necessity, a saddle in between. So this you all recognize. You could, of course, turn that into a landscape, i.e. there's a mountain, there is a valley, and there's a saddle in between. So one-to-one mapping between the two. That you all see very readily. Okay, so in general, we want to turn flows into shapes because there's sophisticated mathematics that has been worked out by Steve Smale and others, proving the Poincare conjecture. And that is exceedingly useful for us, even though in detail I do not understand Smale's proofs. But it's phenomenology, so people, as I hope to show you, biologists in particular can get intuition from these pictures, even though they also don't understand Smale's proofs. So in particular, you know that a glove, I could think of a hand as 5 attractors and 1 repeller, which is my wrist, which is the North Pole. And I could obviously distort the glove into a sphere with the North Pole as the repeller and the South Pole as the unique attractor by doing a bunch of saddle node bifurcations to eliminate the fingers. So that then leads to a theorem that all life is downhill, which perhaps you will regard as intuitively obvious, in particular given David Schwergel's preamble. In any event, there was a theorem due to Smale in 1962, which says that if I define a Moore-Smale system, which is basically a discrete number of rest points, saddles, attractors, sinks, no chaos, and if for the moment I ignore also periodic orbits, and in particular I insist that the system be structurally stable, and that remark is that nearby systems are equivalent in a topological sense. And that remark is very similar to what every embryologist would say, that embryos are robust, you kick them a bit, you will still get a fetus and an adult out the end. So that mathematics is in multiple ways rather similar to basic embryology. Given those assumptions, and ignoring the orbits for the moment, Smale showed that the flow can be written as a metric times the gradient of a potential. That's a very strong statement. It is proved, so you can't argue. At least you have to argue with Smale, not with me. Basically, the flow builds a potential, and to give you some vague intuition, the height of the potential equals the number of unstable directions at the critical point, and from that you could build the rest. That is not obvious. But I note here that's equality, not sort of like vaguely equal to equals. Okay, so therefore a Moore-Smale system for my purposes equals a Waddington landscape theorem. And now we want to ask, how do these landscapes change, which are bifurcations, and how do these bifurcations fit together to make global parameter space? And there was a very short list of events, typical normal forms that allow you to classify everything. And of course, this is now, as regards a set of differential equations, this is mathematics. Obviously, as regards biology, it is not obvious. Whereas how much you can insist that biology follows a set of differential equations is, of course, a matter to be resolved by experiment and data fitting. It is not to be asserted. Okay, anyway, but still with the mathematics, we have a result from those gentlemen that there are two typical bifurcations that suffice to convert any two Moore-Smale systems on the same manifold to each other. And those are the one, the glove to sphere, which is a saddle node, or 1 minima to 2 minima, which you all have seen pictures of. And the other one is rather more interesting, which all biologists implicitly know, which is a progenitor cell in state capital A has a bifurcation at little a, saddle point, and can give rise to one of two possible progenitor states. So it's development 101. Development is basically, as regards cell types, a tree, and this is one node of that tree. Okay. So just to drill this in a little bit, I show you in more detail the topography. So either when you leave A, you go to B. There is an atypical parameter tuned state where this saddle, the outside of that saddle goes to the inside of this saddle, or I could more generally flop over to C from A. So those are the two topographies, and you should keep those in mind since they will recur. Okay. So now the phenomena of dimensional reduction. So without parameter tuning and for generic systems, i.e. structurally stable, there are two types of bifurcations that are typical. One is the saddle node that you know about, and the other one is that heteroclinic flip, which was the subject of the previous slide in those topographies with three stable states. There is an exception to that rule, which is a non-generic, atypical case in which the initial conditions are very isotropic, which have a symmetry built into the initial conditions. And that is also very biological. It, for example, occurred in this data, real data on a real fly, the back of the fly, the notum, developed sensory bristles, and they are spaced roughly 1 per every 25 cells in a vaguely hexagonal pattern. And that process was modeled by these people. And basically, it is one of these examples where the initial conditions, the initial data is symmetric, and there is a symmetry breaking in a relatively large space. If you want to reduce that to geometry, consider a bunch of cells on a ring where there is repression between cells dependent upon distance. And that, of course, is implemented by notch delta signaling in this case. So what happens is, now this is mathematics, you take such a system, and what you see is that there is a high dimensional saddle point where one direction comes in this case, 5 directions go out, all unstable, all symmetrically. And that is the competition between cells to become bristle. So whoever is ahead in the race to become bristle secretes a signal, inhibits others, and the end result of this, for the parameters I happen to choose, was that 2 antipodal cells become bristles, become neural. Everything else is blocked, but which 2 cells one chooses is completely arbitrary, dependent upon a little bit of noise. Okay. So let's get more into biology. A very standard, well-studied Ising model, if I could use those terms, example of a lineage bifurcation is in the early stages of mouse, or in fact human, development, which is very, very similar. So fertilized egg divides. There's an inner cell type and an outer cell type. There's a cavity. And this inner cell type then bifurcates again to give you 2 cell types which have different spatial orientations, spatial positions. And it's the second bifurcation we will study. And then, of course, eventually this structure goes on to implant in the nucleus and make a baby mouse or a baby human, as the case may be. So this inner cell mass, so-called, is positive for 2 markers. The subsequent lineages are positive for a single marker. So there's a bifurcation in types, which, of course, one uses to follow the transition. OK, so a great deal is known about this lineage decision. And we know all the molecular players. And in principle, and in particular, the ratio in the number of the 2 terminal cell types is strictly controlled by the embryo. As you can imagine, it's important for function. And we know what how that works molecularly. The epiblast secretes a signal, a diffusing signal, a protein, which inhibits its source and activates the other cell line. So that, plus a lot of other machinery, suffices to give you 2 cell types. And people have shown in rather interesting ways that the ratio of those 2 cell types is strictly fixed, i.e., you could take the mouse embryo, you could put in cells that cannot become one of the cell types, and the remaining wild-type normal cells will pick up the slack as best they can, sort of like the Simons Foundation and funding. And that was shown here, if I could be facetious about a serious subject. OK, so people have written down models for this with various variables which we don't care about, and they have reduced them to equations which are incomprehensible and have 35 parameters. You can't deal with that. However, it suffices to look at pictures, which are much easier. So here are 2 scenarios whereby 3 states, each of which PE or epiblast is connected to the ICM, the central state, with 2 saddles. And this is topologically one-dimensional. So the transition is that these 2 saddles pinch off simultaneously with the central state and become this. So this picture becomes that picture. And as you could imagine, this is a codimension 2. This is an atypical bifurcation because these 2 things have to move and pinch off simultaneously with the central state. But that can happen, but it requires some parameter tuning. The alternative is the heteroclinic flip. Now the flip, by just drawing this picture, you immediately see experimental consequences. The flip, for example, there is compression along this direction. So even when these two states bifurcate away in a saddle node, there is still compression. So what that means experimentally for an experiment is that the system initially is a valley early on and becomes a ridge, i.e. the two things separate later on. So a valley becomes a ridge. So of course, a valley is rather restrictive for perturbations. The ridge, of course, amplifies. So that is an experimental signature which is immediately obvious to you from the geometry. No fancy mathematics. You just draw a picture and you look, and that qualitative feature comes out, is apparent to you. And that, of course, is grounds, is a subject for experimental checking. Okay, and then we could similarly ask when we use the morphogens. So in this scenario, when we use morphogens to tip things back left to right, do we transit back through this state? Whereas here we don't, once we leave the ICM state, we can transit between this and this, but we don't go via that. So these are qualitative changes which are obvious from the geometry, obvious to an experimenter, and therefore it's a much more powerful way of representing these transitions, not by a bunch of equations. Okay, so we have dutifully made a model for this, and I just tell you qualitatively the ingredients. So we write differential equations for x and y in that landscape. We know from imaging that the cells mix rapidly. Therefore, it suffices to write a signal, FGF, as a function of time, which activates one state, represses the other, and this signal tilts the landscape left to right. And then we have to write an equation for the signal, which is the sum of just polynomial coordinates for that, for the x direction where I'm tilting. The parameters involve, I have to take model time to embryo time. I assume FGF is linear in coordinates, and I have to tilt the landscape in x and y. These are obviously minimal parameters. So this is as simple as it gets. And this model, published down there, explains all existing data. I show you one piece of that data, which is one of these compensation experiments. And it's surprising how accurately the embryo attempts to reach this fixed population ratio that really is tightly controlled by the embryo. So if I put in cells that cannot assume one of the fates, the remaining cells pick up the slack as best possible, given their number. And that was done here, and is a very nice experiment. Very direct, no ifs, ands, or buts, no fancy interpretation, but the model produces it. Okay, but we want to be a little more ambitious. So we would like to now make time-dependent experiments on this system because that's more challenging. And of course, sociologically, you have to realize biologists don't do that because they have no means of interpreting the results. And until they do, and again, as was explained to me by a well-known immunologist at Rockefeller, his favorite experiment is feet up or feet down, i.e. the mouse is dead or alive. That's a very nice, manifestly binary readout, but it is not very indicative of dynamics, being dead or alive. So we want to do experiments that are much more dynamic, and we want to do live imaging. So for example, so we make mice which have a red gene and a green gene. You mate them, and a quarter of the time you'll get a mouse which has two genes labeled. And then the question is, seems seemingly sort of trivial until you think about it. How does an embryo measure time? Is there a timer gene, like a little hourglass, that drains out of the top? And to what extent is the clock, so to speak, gene expression? So for example, with the data I will show you, we could measure cell number, which has been a surrogate for time, how many cells, because cells keep dividing, versus gene expression, which we could see via our live markers. The other thing which has never been done in this system, although it's immensely well studied, is commitment. So commitment means when has a developmental system gone far enough down the valleys so that you can't interchange the two cell types? And of course, we can do that very readily because we're imaging live. We could put on this or that morphogen, this or that drug, and ask when or to what extent can we flip cells from their primitive endoderm valley to their epiblast valley. All very accessible. And then we have to, in this experiment, directly fit the colors because we could map the topography onto the colors via a smooth function. Okay, so I will show you movies of this. So this is one population of cells, the second population of cells, the trophoblast cells here. And you see that at the end of the picture, these are Z slices. You end up with a epiblast, which is green, which is in the top of the sphere, a layer of red, which is more clearly shown here, and you have the two cell fates in a normal embryo. We can also look at how cells specialize. So we have cells that are initially down here, double positive cells, expressing both markers, and then as a function of time, those cells will spread to the epi column or the primitive endoderm column, and these little worm-like things are basically an attempt to see the dynamics. So they're basically things that are color-coded as a function of time to give you a sense of direction. So they go from blue to yellow is the direction. So there's an occasional lost cell, and that comes back. So in the end, you end up with the two populations. And of course, given an image such as this, we could now put on the drug which favors this state or favors that state and ask, when do the cells cross over the barrier so that they can't go back? So that's all so this is all in progress. I can't show you definitive data, but hopefully you can appreciate the question and the fact that one has the experimental means to answer it. And the rest is, as they say, arithmetic. Okay. How do we build the Waddington landscape for a ventral neural tube using stem cells? Now, I should emphasize, again, for background, that the stem cell technology has revolutionized, I believe, developmental biology, particularly for mammals. So what the situation is that you have a cell type that in a suitable medium can be indefinitely expanded and retains the capacity to produce all cell types in the body. And what people have done to varying degrees is to take those cells in artificial situations and ask what is their capacity with defined environments to do what they do in vivo. So you could basically disassemble, take apart all the regulatory system, be they gene networks or even morphogenesis. So all of these things are being very actively investigated by many, many people. And I will show you just a snippet of that data obtained with the Briscoe lab in Crick and pertains to the neural tube. Now, the neural tube, as in many things in biology, as I am fond of remarking, developmental biology was not designed to simplify the task of the theorist. Developmental biology was designed to be robust, and in this particular context, in various ways, the morphogenesis and the gene regulation are coupled together, because the morphogenesis is a very good cue for how the regulation has to work and has to be concordant. And manifestly, biology uses that cue to control the gene expression network. But with stem cells, we could take all these things apart. So in particular, in the textbook, you will see that the early stages of neural tube patterning from ventral to dorsal, say in the trunk region, is governed by two signaling pathways from the top and from the bottom. And these give rise to eventually 11 types of cells. And for those of you who have thought about these morphogens and fates, this looks very surprising. It's very hard to imagine mathematically how you would get 11 fates out of these two presumably static but not static morphogens. But the system works because we're all here talking about it. So what we will do or have done is to simply take the cells out of context, to let them be sort of isolated on slide, apply suitable morphogen signals that induce them to acquire these ventral fates. And we do that as a function of time and ask, how can we model that? The data that we have to work with is basically, it's a little bit antediluvian, but it's still quite useful. So the experimental setup is that we can read out the levels of 5 genes, which are shown here, just names. You don't care about what the names are. And those 5 genes in combination can distinguish 4 fates. If you just look at the combinatorics there, you'll see 4 fates are distinguished. So that is the readout, and they're proteins, which is good. OK, so what do? We make a landscape in 5 dimensions. So the way we do that, and I skip over technical details, but you could all imagine how it works. We first of all have to take, we have 1 million cells. We sample them in equal numbers for each morphogen level and each time window that they sample. And we then ask, we pull all the data, and we ask where there are local maxima in the point density in 5D. We then, but of course the real data is not static. The real data is flowing downhill from the progenitor state to the various terminal states that were in the previous slide. And we want to model that flow downhill. So the way we fit data is to take those local minima in density, which I will show you have state names, biological names attached to them, and then we simply use as fitting parameters, we raise the height of those minima. It's a potential, right? Potential, it's what you do with a potential. So you raise the height of those minima, and then we have to interpolate, so the fitting parameters are the heights, and then we have to interpolate between them, and we do this, which is standard computational vision. And then we fit a path integral, which is time rate of change in 5D is gradient of potential plus a bit of noise. And that's a path integral, which you all understand. Or Feynman-Katz path integral, if we want to make a genuflection in the direction of mathematics. OK, so that's what we do. So why is this a useful strategy? First of all, we have a potential. We fit a potential. Therefore, we can compute eigenvalues and eigenvectors of the potential. So saddle points have one unstable direction. That's a result. It's not an assumption. Fixed points have all stable directions. That's a result, not an assumption. Most importantly, which you may think is trivial, but it's actually rather profound, is that we can interpolate by interpolating potentials. Now, you may say it's obvious, trivial. You have Arrhenius. But in fact, if you were perhaps a biologist, you might try to interpolate densities. And that's a disaster, because one is related by an exponent, and taking the exponent and taking the sum in an exponent is not taking the sum in the output. Very different. So we get actually rather profound results for free from the formalism. We also we can infer a model for any time course you care to throw at me, because we fit the data for fixed morphogen levels. And then if you tell me you're switching morphogens in time, I can compute what should happen based on this constant time fitting. And then we displayed data with diffusion maps. And diffusion maps you probably don't care about. It's basically look at the eigenfunctions of the Fokker-Planck equation. If you recognize that, if you don't, it's immaterial. OK, so what do we do? So this is the graph of all the densities, all the cells superimposed to ask where are the states of high density. So for example, the state P3, which is of high density, is defined by saying what markers are high. So this state P3 has high SOX2, high NKX 6.1, and high NKX 2.2. Those triple of markers define P3. So P3 is high this, high SOX2, and high 6.1. So all of these minima that we find in the data correspond to states in the neural tube as defined by gene expression. So there are no phantom states. Okay, so we just look at the FACS markers. Then we fit data. So here on the top row is experiment. So we're given data every day, and we're given a cloud of points, which I'm projecting onto 2D. We're given a cloud of points, and the cloud of points evolves from the initial state. This is high morphogen. So it evolves from the initial state, moves along the top of the picture, moves to the corner, and then some of it moves down here to P3. So that's the data we are given for a fixed morphogen level. We could then fit that data by the process I showed you, i.e. controlling the heights of these various minima, which are fitting parameters. And we could fit, so this is the fit. And the starting data has 12 bits of information. The fit has relative information of 0.3 bits, so we're getting 97% of the variance, which for biology is very good. Right? This is not atomic physics, regrettably. 97% of the signal is very good to capture. So we're capturing things very well. We see, now I show you the dynamic potential that resulted in those fits. So here is my crib sheet for the states. Here is the mapping to the embryo. So for example, we see at low morphogen level that the data begins, the states begin, the cells begin here, then they go downhill to this state, which is P01, and they somewhat move over to P2. They are blocked by high potential along this left edge. So they just move along here and they stop here. For a high morphogen level, you see these early states become high potential, so things run downhill from initial state to P01 to P2, and they keep going downhill. Some of them land here, and some of them go on to P3, and there's a saddle in between. So this is the potential for high morphogen, where obviously cells have bifurcated away. And for high morphogen, you see that we're occupying states here, and for low morphogen, you see we're occupying states over here, which sort of makes sense. But the potential gives you the dynamics in the 5 dimensions. And we're fitting dynamics, and of course, we can discern the potential only because we could fit dynamics. So that is all tight in 5 dimensions. Okay, so what do we conclude from all this? We conclude that the transitions, which I claim as a result, go from sampling this space, sampling these states, to this state, to this state, to this state in time. I haven't showed you in detail, but we have seen that you could accurately map, you could accurately interpolate the potential. So for middle SAG levels, sonic hedgehog levels, we can interpolate between 10 and 500 and very accurately fit the data. That is not obvious. So that's the result. The other thing is that we could generate a null model for various time-dependent protocols. Those fail badly. But in a quantitative context, failing badly is actually rather informative because in this case, we know that there is basically chromatin memory, and a small exposure to high SAG basically changes your chromatin. There is memory, and you can't drop down in morphogen level. So there's a ratchet effect, which we see because the time dependence fails. You can't unwind these potentials because, again, and here we know the example is due to chromatin. Okay, so that is very roughly speaking conclusions. How do cells make signals influence other cells? So how can we think more about cell interactions and tissues and so on? And in this case, I point to some old work which still has interesting lessons to teach us. And that's a very, very classical textbook system of the vulva in the worm. And here we have a situation where there are either 5 or 6 cells which are an equivalence group. An equivalence group means that any subset of those 5 or 6 can make the desired organ. So that's example, again, canalization. You want to make the vulva for mating purposes and egg-laying purposes, and if you don't, you're obviously very dead. So there are 5 such cells, and some very classical genetics. And when you think about this as a quantitative person, it is quite remarkable that somebody staring at worms, cells, vulva cells dividing and their division patterns can infer much of what we know about the Notch delta pathway. That is truly remarkable if you think about it. I mean, I couldn't do that. But people literally spent years and years and years staring at these worms to great effect. Anyway, so we could take that data, and we know that the symmetry is broken by an external cell called the anchor cell that secretes an EGF signal, and that EGF signal reacts back on the central cell via autocrine signaling, and then it induces the more distant cells by a combination of EGF and Notch delta. So we have to model how does the Notch delta signal from this cell, how does it turn on and influence those 2 other cells? And I should still have, okay, let me do this because I do have a moment, and it's worth seeing, it's worth seeing the movie. I need my glasses. I ask for your forbearance, and I need my glasses. Okay. Don't skip slides. Sorry. This is actually worth seeing. Okay, so now we go back to okay, so here is a movie, thank you, of those 3 cells. This is a model, but the model fits a lot of data. So the cells are initially pre-competent. They do not see the signal. They bounce around in their respective minima, which is the same. This is an equivalence group. Now the cells see the morphogens. And as this central cell moves towards this line, it emits a Notch delta signal and changes the topography of the adjacent cell. And then finally, cells no longer see morphogens, and everything goes back downhill to its respective minima, and this is the wild type, i.e. normal pattern, red, green, blue. Okay, that's just this is all published, so I don't dwell on it too much. But the thing I wish to emphasize and underline is that this particular model explains a lot of gene epistasis. Gene epistasis is interaction between 2 genes, so that if you understand gene 1 and you understand gene 2, and you put them together, you can't quite guess what the double mutant does. Now, this particular model is mathematically linear. That is to say, it is, in terms of fitting parameters, there's a parameter for each gene or each mutation, and that's it. The nonlinearity comes about from the mathematics. And that nonlinearity, basically, then what we have is we could fit a vector of parameters, and we could make a matrix of predictions. That's very potent. And those predictions work as shown in these previous papers, and also in a new paper from this lab in Paris, which does dynamic measurements using temperature-sensitive mutants. So you could change the temperature, turn on and off genes, and see what happens in a time-dependent way, imaging. And that's very potent. But just qualitatively, the content of the model is that when you have initial conditions that place you near a saddle point, and a saddle point to a geneticist is a sensitized background, which you do because your new gene, your new mutation, doesn't have a big effect on the wild type cells, but if you put it in a sensitized background, you'll see something. And people try to infer what that something is by what happens. So here, for example, but being on a sensitized background is, in mathematical terms, being on the inset to a saddle point. So you're near a decision point. And when you're near a decision point, in fact, there were experiments, incorrectly interpreted in our view, which took this situation, pushed it to the right, we would say, and they got an outcome shown here in which the cells go up to the P2 fate. So you push this way, and your result is that way. It was interpreted in the experimental paper that this signal, when weak, pushed up, so that they interpreted the red as being like the green. That is not the case. We can explain their data with this manifestly linear model, and the phenomena has to do with the fact that it's a nonlinear system, right? Everyone knows that. It's a nonlinear system. You cannot infer from this initial push, you cannot infer from the outcome that the initial push was horizontal. It's a nonlinear system. And that, in this model, that nonlinearity is built in through the model. So it comes out as a consequence, and you can make lots of predictions, and they have been checked both on old data and then also here in new data. So that, I claim, is potent. OK, so where does this all leave us? OK, so genetics, so a good genetic screen is very much like my immunologist feet up, feet down. It's a clean endpoint that you could quickly score and is black and white. Those experiments in many different systems have given us the parts list, but it's a very awkward experiment in which to use, to which to employ to fit dynamics. You really want to look to do experiments that perturb the topography as things are going downhill, that look at the saddle point. So you actually prefer outcomes that are shades of gray because they show that you are sitting on the boundary, and you want to define boundaries. You don't want black and white outcomes. You want muddy outcomes. And to interpret that stuff, you need a model, because the experimenters can't just look at the data and say, aha, this is what's going on. You need a quantitative model because it's not obvious to anybody. So mathematics enumerates a short list of models, so you sort of know what to play with, and it then behooves you to try to fit these things. And the modeling depends very much on when you apply the signal as much as what you apply. This work was done with a group of people who are almost ergodically scattered across the globe, as you can see here. And the experiments have come from people at the Crick, people at MSK across the street, and also from Paris. And with that, I leave you with a word of the day from Darwin. I'm a firm believer that without speculation, there's no good in original observation. So keep that as your motto. Thank you. Lots of time for questions. Yes?
Thank you for the very nice talk. So I'm wondering, in your sort of gradient flow formula,
Yes.
when things are flowing downhill,
Yes.
particularly in the context of like time-dependent morphogens,
Yes. They flow downhill even without time-dependent morphogens. That, of course, changed. But I mean, you know, the initial state is unstable to these developed states, and they go downhill in the appropriate media.
but in the presence of time dependence,
Yes.
is there any assumption of like quasi-equilibrium or?
OK, you're assuming, fine, that's obviously very important. You are assuming, or you can do anything you like, but I mean, you have to say when the experimenter changes the media, to what extent is there stuff that's still stuck on receptors, stuff that's being internalized on endosomes, going to the nucleus, so you're making an assumption, obviously. And of course, you could, in the computation, fiddle that time lag. But what was the reason that that did not work here is that we got them to do the right experiment such that even if you're in the high morphogen state for very little time, you're really stuck. But you could really insist upon that and pound the table when you have a quantitative model which shows what the data would look like if you could go back. So it's very useful to have a quantitative model say, if you could reverse the topography, what would happen to the cells? And the result was so far from being plausible in that context that we say, and it was known that there are obviously ratchet, so to speak, chromatin effects. You know, when in doubt, blame the chromatin, as they say. Yes?
How does this work? Can you hear me?
Yes?
Okay, so regarding commitment and irreversibility,
Yes.
So right now we have experiments that show that you can differentiate cells and
Yes.
bring it back to past states. So
Yes.
some
Yes.
states that were previously considered to be committed
Yes,
are actually pretty reversible
Correct.
given
Correct.
conditions. So
Right.
does that mean that the Wellington landscape actually changes? And
Okay, yes.
how would you explain that a valley would
Okay, very
disappear
good.
and reappear depending
Right,
on certain
first,
conditions?
That's a very interesting question. So for the others, you know that Yamanaka in Japan got his Nobel Prize for taking differentiated skin cells and bringing them back to pluripotent by overexpressing four transcription factors, Yamanaka factors. Other people have taken smaller steps, always going backwards. The rule of thumb is going backwards, you get 1% yield or something. You get very low yields. So, so it's very interesting. So again, I think of that very schematically, but not quantitatively, as basically trying to find, you're in a complex landscape, you're trying to find some valley backwards, and there's an entropy cost to finding the valley. So there's some sort of loss of yield. Also, when people do dedifferentiation, the time scales, the cells go through the topography backwards in a very heterogeneous way. And people have not really sampled that in detail. It is also true, and there's data from my collaborator, Kat Hadjianakis, pushing backwards a extraembryonic endoderm-like cell line, Zen cells so-called, pushing them back to pluripotent. And in that case, they know that the path backwards is not the same as does not retrace the path forward. It's a small enough jump, and there's enough markers they have. So all that is very interesting. So the cells, but you're forcing the expression of transcription factors. So you're obviously, by forcing that, you're changing the topography in some way. But it's very interesting to ask, as they did in that case, whether you retrace your steps, and you don't. You take a shortcut of some way, and that's rather interesting. But again, I think a topographic analysis is interesting. And of course, you're going over that topography in a very temporally asynchronous fashion. Yes? Push your buttons. Yeah, you have to be.
Can you hear me?
Yes.
Okay. So this is not my field at all.
Yes.
And you mentioned, all this stuff seems to be fixed points and gradient systems and no limit cycles or anything.
Yes.
But my understanding is things like somatogenesis depends on rhythmic phenomena.
Yes,
I'm
very
wondering
good.
how that would fit in
Yes,
with this.
very good. Yeah, we spent a lot of time thinking about somatogenesis, precisely because it's how you go from a periodic phenomena to actually the somites are spatially patterned. So how you go from so the Morse-Mel systems technically allow limit cycles. Obviously, it's not potential, but even in the case of limit cycles, there is a Lyapunov exponent. That's a paper by Mayer a few years after Smale. So somatogenesis is we still would use geometric methods to ask how does a torus breaking down give rise to basically a binary pattern. And for example, you might think about SNCs, saddle node on a circle. And in fact, in somatogenesis, you see an effect of lengthening periods just prior to making the somites, which is an indication. But there's also other phenomena. I could give an hour lecture on somatogenesis, which I won't. There's another phenomena which is an anticipation, a pre-pattern is established several somites before the visible somites. And the question is, what is causing that pre-pattern? It is not clear genetically. It is potentially structural. But there's very fragmentary data on that. But certainly the question is very relevant. It's amenable to geometric methods because you know how tori break down. And it's very interesting. Yes. We shouldn't privilege people in the center section, though.
You showed that in the neural tube that with gradient of morphogens
Yes.
that you can access all those 5 different states.
Yes.
What is the biological, relevance?
Yes.
If you use too much of the morphogens,
Yes,
are you
okay.
getting a good representation?
Right. It is a very valid question and deserves to be underlined as to limitations of the effectively single cell systems that ignore all the cell communication that we know must go on in the embryo. So there is particularly in other systems, maybe not in mice, spatial cell sorting. There is also Notch delta signaling. Lots of things are going on. So those are basically being eliminated in these single cell approximations. Their virtue is that you could ask, and of course the actual morphogens go up and down as a function of time. And we don't know what effect that has, although it can be imitated. But again, the interest of the stem cell systems is that you could take apart the regulation and say, from the morphogen only what happens, and how much of that is reality, and how much is not. So it's basically, it's a poor excuse for a real system, but it allows you to deconstruct the signaling. The other thing that should be said is that we could also computationally play with the noise. And you noticed, if you looked very, very carefully, that as a function of the morphogen level, we never got pure populations at any morphogen level. And we think, and that's in the data, right? The data didn't. We think that is because this artificial system enhances the noise, i.e. it ignores environment and the cells obviously see each other. So what we did, just as a game, a thought exercise, is to lower the noise level in the model. Then we could very readily get pure populations at various morphogen levels, suggesting that the artificial system is much too noisy. But that's, of course, a speculation. But it's extremely useful. And when the morphogen really goes out of bounds, you get floor plate. And there's other things we've played with to show structure, spatially and structurally how that occurs. So, but again, the thing to carry with you is that these synthetic systems can also self-organize in 3D spontaneously. Imperfectly, to be sure, but it is extremely interesting to see how well they do, right? As Sam Johnson once says, it's not done well, but it's surprising to see it done at all. And that is indeed true, and it's very instructive. Other questions? Yes, in the back. Your button.
So, to your point about understanding the geometry of the flow as a fundamental entity,
Yes.
I totally agree. 35 couple differential equations aren't going to give you much insight.
Right.
But then what it makes me wonder is, do we need the model, the math at all, or do we just need the geometry? Can we just try to infer the geometry of flow directly from the data, and then what do we get by adding the math?
Okay, very good. Good. That's, again, a very germane assumption. It is, okay, so the interesting part of that question has to do with how we deal with morphogens that saturate, right? So we all know that at some level you're at maximum response. So the question is, how do you write down, and if you just do a naive normal form, you have parameters. And if those parameters were thought of as linear, then of course a factor of 10, which is sort of the experimental range, knocks you out of contention. So the more subtle part of the modeling is to actually understand phenomenologically how to saturate it. And in particular, but again, the problem has, the desideratum has to be, having done that in some way, can you predict gene epistasis? So for that Vulva model, we rather elaborately showed that various sorts of gene interactions that were in the literature could be predicted by a manifestly linear model. That is non-trivial. So that is, and that is above and beyond naive normal forms, because we in a rather clever way saturated the model. So the fact that as a first approximation you could put the potential into the exponent is a way of saturating, because in the exponent, things that are off scale give you 0. So that very simple Arrhenius form actually packs potent import in these contexts if you're willing to think about things as a potential and path integral.
Okay.
Good, I've exhausted you. Oh, yeah, sorry, one more.
So, I have two naive questions. One is,
Louder.
So, you mentioned, you have noise as, the factor that decides for you,
Yes.
at the, decision points.
Yes.
So I'm wondering, what kind of noise you have in the system?
Okay, very good. Yes, also a very good question. The noise is simple additive noise.
Okay.
If you look at what the point clouds look like, particularly in that Vulva model, the point clouds are very much shaped by the saddles, by the stable and unstable manifolds of the saddles. So as a function of time, very quickly, the distribution of points is highly asymmetric. And that's a consequence of the dynamics. So we did not need to fit data to do anything fancier than isotropic noise.
Okay.
But the topography then very strongly shapes the point clouds, as you would expect. You know, you're moving in a valley or a ridge or something.
Sorry, second
Yes.
naive question, if I may. So,
Yes.
like you mentioned, like you have a model of like very lot of parameters. I wonder like if we really need that much parameter to do the fitting, or like have you tested something like if you have like a little bit perturbation to the model and you
Yeah,
try to
okay.
fit it into the data?
Right,
does
okay,
it,
so
like-
you could ask us more technically, can we compute the Lyapunov, the Jacobian of the fit with respect to the parameters?
yes.
And that shows, when we do that, it shows we don't have very soft directions. Intuitively, for the, say, the Vulva model, we basically have one parameter per gene pathway. And of course, if you're hitting whatever the FGF-ERK pathway, there are many points where it's a linear, sort of a linear pathway. So you could hit at any points, they influence the same parameter. So that condensation we see, but, and indeed for the stem cells on topography, we're simply raising, the minimal thing is simply to raise the heights of the potential. You can't do anything simpler.
I see.
And that suffices. Again, it's not very high precision, short time data. But given the data, that's as good as that
I
is
see.
plausibly sufficient.
Thanks.
Okay, good, break, I guess.